Transcription is a process by which genetic information on DNA is copied to form mRNA. There are 3 stages in transcription. Stage 1. Initiation. Transcription starts at a specific site on DNA called promoter. Different genes have different promoter. Promoter act as starting point for transcription of a gene, and it provide the binding site for RNA polymerase. When RNA polymerase binds to promoter, RNA polymerase unwinds the double helix DNA and can initiate mRNA synthesis. Stage 2. Elongation. The portion of the DNA that is transcribed into RNA is called the transcription unit. RNA polymerase moves along the template DNA strand. RNA polymerase only adds RNA nucleotides to the 3' end of the RNA transcript chain. Transcription occurs in the direction of 5' to 3', forming a single strand of RNA complementary to the DNA template. Cytosine will pair with guanine, and uracil will pair with adenine. Behind the point of RNA synthesis, the double helix DNA reforms and the RNA molecule peels away. Stage 3. Termination. Transcription stops at a specific site on DNA called terminator. Once RNA polymerase has gone past the terminator, the RNA transcript, which is pre-mRNA, is released. RNA polymerase detaches from DNA template. Transcription will produce pre-mRNA first before mRNA. Pre-mRNA contain introns that lie between exons. Exons are the coding segments, translated into amino acid sequences. Introns are the non-coding segments that are not translated. During RNA processing or RNA splicing, first, introns are removed. Exons are joined together forming mRNA. Spliceosome is involved during this process. mRNA contains only exons and ready-to-leave nucleus to be translated in cytoplasm. Translation is a process where codons in mRNA is translated into a sequence of amino acid to produce polypeptide chain. Translation occurs in cytoplasm. There are 3 stages in translation. Stage 1. Initiation. Initiation involves start codon, AUG, that encode for amino acid methionine. Small ribosomal subunit binds to 5' end of mRNA. Start codon signals the initiation of translation. The initiator tRNA with anticodon UAC, which carries amino acid methionine, binds to the start codon and followed by the attachment of large ribosomal subunit forming translation initiation complex. The initiator tRNA is in the P site of ribosome. The empty A site is ready for the next amino acid tRNA. Stage 2. Elongation. There are 3 steps in the elongation of a polypeptide chain. Step 1. Codon recognition. Next amino acid tRNA binds to codon into A site by complementary base pairing between the mRNA's codon and the tRNA's anticodon. Step 2. Peptide bond formation. Enzyme peptidyl transferase catalyzes the formation of a peptide bond between carboxyl terminal at first amino acid and amino terminal of second amino acid. The growing polypeptide chain of tRNA at P site is transferred to the amino acid of tRNA at A site. Step 3. Translocation. The ribosome translocate tRNA with the attached polypeptide from A site, now at P site. The empty tRNA in the P site is moved to the E site where it is released. The A site is ready to bind new amino acid tRNA. The process is repeated until complete polypeptide chain is formed. Stage 3. Termination. Termination involving stop codon, UAA, UAG, UGA, which are act as signal to stop translation, and they do not encode for any amino acids. When a ribosome reaches a stop codon on mRNA, the A site of the ribosome accepts a protein called release factor. Release factor hydrolyzes the bond between tRNA in P site and the last amino acid of the polypeptide chain. The polypeptide is released from ribosome. Ribosomal subunits and other components dissociate.